Good morning. I will be presenting here the ICCP Global Mammal Model. So here we have a global dynamic model which simulates the biomass distribution of all the mammal species around the globe in response to the climate forcing. A quick look on the terrestrial mammal distribution. Here, the wild mammal species, their contribution to the total biomass is very small, and the contribution of wild terrestrial mammals is even smaller compared to the human biomass and the domesticated biomass. But they have a significant role in the food chain, and they are distributed all over the globe. Here on the top right is the estimated total biomass of present day mammals, and you can see that the total biomass is high over this tropical rainforest where the net primary productivity is high, and the corresponding species richness is also high over these regions where the biomass and the diversity is high. And these are regions where the vegetation is thick and diverse, and here comes the role of climate. And any long-term change in climate can result in a decline in biodiversity and can lead to extinctions. And we have seen this in the late Quaternary period. On the left panel is the global mammal biomass along with the human and livestock, and after the mass extinctions in the late Quaternary era, we can see that the wild mammal biomass has reduced significantly. And on the right panel is the mammal extinctions as a function of body mass. In the lower body mass ranges, the relative number of extinctions are lower, and if you go to the higher body mass ranges, the megafauna, the extinctions are relatively higher, including this iconic species like the mammoth. And what caused these extinctions is still a question, whether it is climate or humans or both, and why the megafauna. And from these archaeological evidences and paleoclimate processes, we have some hint on the role of climate and humans. The climate-induced extinction risk is more prominent over these extratropical regions where we saw the extinction of the species like mammoth. And looking at the time evolution of human population and the number of megafauna species, we can see that the dramatic drop in megafauna species coincides with the human arrival. Now we need some tool to separate out the effect of climate and humans. So we need modeling techniques, and here we have a model. Our model basically uses this reaction diffusion equations into which the climate forcings and the mammal trait data we use as input. The climate data includes the temperature, precipitation from which the net primary productivity is estimated, and we also use the mammal trait data including the body mass and the trophic level. From the body mass, we use the body mass to scale the mammal characteristics, including the growth rate, lifespan, diffusion, etc. And the climate data is used to estimate the habitat suitability of each mammal species, taking into account this occurrence data set. And all these inputs and forcings goes into this partial differential equation for each mammal In total, we have about 2000 mammal species in our model, and finally we get the biomass for herbivores, which feeds into the biomass of carnivores. These two trophic levels interact through predation in our model. This is a quick look on the body mass of mammals that we use in our model. In total, we have about 2000 mammal species, about 130 carnivores, and about 2000 different herbivores, ranging from rodents to bison to the largest mammals, including the mammoth. Here we run a hypothetical situation where we include both the present day and extinct mammals together to see the climatic the response to climate in time. And here is the occurrence data that we use for all the mammals, the red ones for carnivores and green for herbivores. The occurrence data basically includes the fossil data from Pascalet and the other occurrence data from GBIF. This is a summary of our equations. We have two trophic levels in our model, herbivores and carnivores, and here is the time varying biomass tendency for herbivores and carnivores. Here we have a diffusion term which estimates the dispersal of each mammal species based on their population density gradient. And here is a logistic growth term. The logistic growth is controlled by the carrying capacity. For herbivores, the carrying capacity is a function of net primary productivity, and for carnivores, it is a function of the total prey biomass, that is the total biomass of herbivores available for that particular carnivore. And for herbivores, there is a predation term which is a negative term, which basically reduces the biomass per area if there is predators with a habitat overlap with a particular prey. And this logistic growth goes like this. It's an S-shaped curve. In time, when the carrying capacity is high, there is an accelerated growth rate, and then the growth population size reaches a maximum, and when the population size approaches the carrying capacity, the growth starts decreasing and it becomes stable. And here the growth term, this mu, and diffusion term is scaled using body mass. This is an example. Each mammal characteristics can be scaled using their body mass. And we also have species interactions inside our model. We have predation and competition. For that, we use these two matrices. This is the predator-prey matrix. On the x-axis, all the herbivores, and y-axis, all the carnivores. And this blue shade shows the possibility of predation. For that, we use a body mass window, the highest prey mass and the lowest prey mass. And these red extensions shows the enhanced prey availability due to sociality, because some of these carnivores, they prefer solitary predation, but some, like the lion, they prefer social predation, which allows them to chase a bigger animal. Similarly, the competition matrix on the x-axis and y-axis is the species body mass, and this window shows the possible competition. Here also we use a body mass criterion. And I will show you some of the snapshots from our model. The model was forced with climatological forcing for the present day. In the upper panel, 3 selected species, mammoth, reindeer and the canis species. The lower panel is the range maps from phylocyne. These range maps and our model basically uses the same occurrence data, but the difference is that we have this spatial variability in habitat suitability because we include the climate data also in our calculations, which is very important. And the corresponding biomass. The biomass is high over the regions where the habitat is suitable. This habitat values ranges from 0 to 1, where 1 shows high habitat suitability and 0 shows no, the habitat is not suitable. And here is the distribution of biomass for different species and the spatial variability that we obtain from our model. It is very important in finally calculating the biodiversity of around the globe. A comparison of the total biomass from our model here with the observations on the right panel, and here herbivore biomass and carnivore biomass are shown separately. This total biomass, as we have seen before, is high over the tropical rainforest, similar to the observations, which is also consistent with the net primary productivity. So basically the resource availability determines the total biomass distribution, and the interaction between species through competition and predation modulates that magnitude of biomass. And here is a comparison of the contribution of major taxonomical groups to the relative number of species, individuals and biomass. The left panel from the model and the right panel from observations, estimations. So we need to check that other than climate forcing and NPP, the species interactions are also happening within our model. So this final relative distribution of biomass or individuals in each body mass range or the taxonomical groups shows the whether our model is simulating well the species distribution. The relative number of species, if you look at the relative number of species, the rodents have the largest contribution. They are using around 4,000 species, 5,000 species, and we are using about half the number of species because we excluded some species due to the data putting a criterion depending upon the number of occurrence data points. If the occurrence data is not enough, we will just filter that species. But still we are getting a decent distribution. The number of individuals and the number of biomass. This our model captures this distribution very well, even if we are using only lesser number of species, because these species mainly contribute to the total biomass and density. And finally, and most importantly, the species richness, which is the number of species per grid cell or area. Here is the species richness distribution, spatial map from IUCN and from the model, the top panel. The general spatial distribution of species richness is captured by the model, but we have an overestimation of species richness because we are including both the present and extinct mammal species. But we can see this high species richness over the tropical rainforest and lower species richness over the desert regions and other arid regions in the extratropics. So in summary, our model provides a good representation of the terrestrial mammal distribution, and this opens up a new window to explore the relative role of climate and humans on the mammal distribution. And we hope that this will be an effective tool to monitor and predict the future mammalian extinctions, and this can be used for developing the conservation strategies. Thank you.